Hello, everyone, and welcome to a very special Halloween week edition of Lunch with a Scientist. I'm Leah from Headwaters Science Institute, and I was just about to start eating this pumpkin, but I'll put it down for now. Today, we're going to meet a scientist who's going to talk about some ooey gooey creepy frogs. In all seriousness, we are going to meet TJ Firneno, a wonderful scientist who will tell us more about his research. But before we get into that, I wanted to share with you a couple current events. If you haven't seen them on our Facebook or YouTube yet, we're doing 3 Halloween science videos this week. We did one yesterday on how to make monster toothpaste. Tomorrow, I'll teach you how to make edible worm and dirt cups. And on Friday, you can learn how to make pumpkin slime. All of these activities can be done with ingredients you probably already have at home, and they're designed to show you that science is everywhere. So we hope that you have a good time joining us. The second thing I want to share with you is our upcoming movie premiere. The film Picture a Scientist will be shown November 5th through 7th. It's a free screening. All you have to do is register with your email so that we can send you the link to watch. The link to register, it's a short link. It's bit.ly/HSI-science film. It's a great film about gender equality in science, and we think you should be having these kinds of conversations. So please join us. Now to turn to our talk today. TJ Firneno is a 5th-year PhD candidate in the lab of Dr. Matthew Fujita at the University of Texas at Arlington. He obtained his master's in biology at IUP, Indiana University of Pennsylvania, in 2016, and worked in the lab of Dr. Josiah Townsend there. His research focuses on the microevolutionary processes that drive diversification, promote biodiversity, and lead to the evolution of novel traits in organisms. He primarily uses amphibians and reptiles as a model of these organisms to do so. So I am so pleased to welcome TJ Firneno. Hi, TJ.
Thanks for having me.
You're very welcome. We're very excited to talk with you today. If you know a little bit about Headwaters or you've been following us, there's an interesting connection here. TJ has followed a very similar educational path to one of our instructors, Daniel Dudek. Is that correct?
Yep.
So you went to the same school for your master's and are now in the same school for your PhD.
Yep, with the same advisors.
Quite an interesting connection. So if you saw one of Daniel's talks very early in the series about frogs, you're going to learn more about some other frogs today. So TJ is here to talk to us about the processes that taxonomists and evolutionary biologists use to assess biodiversity and to describe new species in the context of a recently newly described species of frog from Honduras. So without further ado, TJ, let's let you take it away.
Great. So thank you all for joining me today, and thank you to the Headwaters Science Institute for having me for this. I'm really excited. And what I'm going to talk to you guys about is basically a snapshot of some of the research that I do that focuses on biodiversity discovery. And I'm going to do this kind of in the context of a case study of a discovery of a new species of microhylid, or a type of frog from Honduras that we recently described. So first, I just want to touch on some of the ideas of what biodiversity discovery actually is. So what it is it's basically an effort to discover and document and describe the different flora and fauna, so the plants and animals that we see on Earth. And really, this is so important right now because it's basically essential for measuring and preserving or conserving biodiversity, especially in the face of anthropogenic change and actually high levels of biodiversity loss, as well as testing biological theories, and also kind of almost as broadest is understanding how ecosystems themselves function based on the biodiversity that we find within them. And you can see here on this slide, in this picture, the kind of vast amount of biodiversity that we see all over the Earth, yet this slide just basically barely scratches the surface of the vast amount of biodiversity that we have on Earth today. And so, in a sense, there are kind of 3 major types of biodiversity, kind of going from the most specific up to kind of your broadest. And Your most specific type of biodiversity that you see is genetic diversity. And this is basically measuring and assessing the amount of variation or variety of genetic makeup within a species or within a group of species. Then we kind of jump up to kind of your middle ground generalization of diversity, which is species diversity. And it's, again, measuring and assessing the variety of species maybe within a given area. So saying within this location, what type of species do we have and how many of them do we have within that location? And then at the broadest type of biodiversity, we have ecosystem biodiversity, which basically is the variety of habitats, ecosystems, communities, et cetera, that are found within certain locations. So again, kind of being able to assess all these different types of biodiversity is super important in the long run to answer big evolutionary hypotheses, again, conservation questions, things like that. And what my research tends to focus on are kind of the most specific, so genetic diversity and species level diversity. And what I'll kind of talk about today is mostly genetic diversity to be able to assess and describe a new species. So kind of starting off, where do I study, or where do some of my research take place? And one of the hotspots for my research is actually Central America. And one of the reasons why Central America is kind of a really great place to study biodiversity and to discover biodiversity is because there are high levels of biodiversity, primarily due to its tropical location. It's kind of moderate climates, tropical climates, allowing for breeding to happen all year round, long, quick generation times, things like that. Therefore, biodiversity tends to be much higher. But also, kind of on the flip side of that, Central America is actually one of the places that we see some of the highest levels of biodiversity loss, primarily due to deforestation in this region, but also, and I'll talk about this later, due to some diseases that have popped up and kind of caused a lack of biodiversity or a loss of biodiversity. And more specifically within Central America, I study or focus on a region called the Tortille Block, which is this area in Honduras and El Salvador and Nicaragua that you can see highlighted here. Hopefully my cursor is showing. And so you have the country of Honduras here, El Salvador here, Nicaragua here, and then outside of the Tortille Block, you've got Guatemala, Belize, Costa Rica, and Panama here, just to give you some context as to where we are. The reason why the Tortille Block or the Tortille Highlands is very interesting to study is that it actually is this kind of mosaic of highland areas. So you can see these highlands where kind of the highest elevation is shown by these kind of red colors, and you can see that they're kind of scattered throughout, along with some slightly lower elevation yellow areas. But they're this mosaic throughout with these large lowland valleys in between them. Basically, you can see those in the dark and light gray here. And what that mosaic does, or that mosaic landscape does, is it actually can promote high amounts of species endemism or specificity, species specificity. That's a great way to put that. In these specific highland locations. And that's because these lowland valleys don't allow for species or organisms that are highland adapted to be able to cross those lowland barriers. So you have high amounts of diversification happening due to this topography of this region. And this region also harbors a really diverse herpetofauna. So as Leia mentioned, I tend to study reptiles and amphibians, and that's what a herpetofauna is. So there's a lot of reptile and amphibian diversity in this region. And kind of getting into, as I just mentioned, kind of what do I study? In terms of the organisms that I study, I tend to focus on anurans, which are a type of amphibian. And these anurans are frogs and toads. One of the reasons why, or a couple of the reasons why they're super interesting to study is that they are actually one of the most diverse vertebrate lineages on Earth, with over 7,000 species of frogs and toads within this group, and still more being described basically daily. They also have very complex natural and life histories, which allow many different hypotheses to be formed based on these organisms, and then tested further to discover specific ideas about these organisms and how they interact with the ecosystems that they're in. They're also keystone species to a lot of the ecosystems and habitats that they inhabit. And what a keystone species is it's an indicator of kind of the health and well-being of the habitat that they're in. So, their frogs and toads are very sensitive to pollutants in their natural habitat. So if the pollutant is in the water, it can affect their tadpoles or the developing eggs that are laid in water. Also air pollutants, things like that. So they're they're key indicators to kind of the health and well-being of the ecosystems in which they inhabit. And kind of on the saddest side of all this is that they're actually becoming highly imperiled due to anthropogenic or climate change, habitat destruction, and disease, which I'll kind of talk about a little bit later in this talk. So all of these kind of characteristics of frogs and toads really make them interesting and good model, kind of non-model model organisms for us to study these different things concerning evolution, systematics, things like that. More specifically, and in the context of what I'm going to be talking about today, the study that we're going to be looking at focuses on a group of anurans called microhylids. And you can see kind of the diversity of microhylids just within this slide is pretty fascinating. And this doesn't even, again, scratch the surface. And they're a relatively speciose group of anurans. They also have a primarily fossorial lifestyle, meaning that these guys spend most of their life underground, burrowed away, and then they come out during really heavy rains to breed and to eat. And it's for a relatively short period of time, maybe a week or two. And then they go back underground and they just stay there for till the next really heavy rains when they can come back out and breed again. And they're also relatively small bodied. So as you can kind of see, these things are on leaves or rocks or things like that. And they get maybe only up to being about the size of a $50, just a little larger than a quarter. So they're relatively small. I mean, when you think about this, they're about that big. So relatively small species, which in terms of studying morphology or the external characteristics of these animals, which a lot of taxonomists do, and I'll talk about that a little bit more in a second, it can make it very difficult because the characteristics that we're trying to look at morphologically or externally, when they're super small, they become much finer to be able to try to tease apart. So microhylids, where there's been a lot of discovery of different species, there actually probably hasn't been as much discovery of species because of how difficult they are to tease apart by external morphology because of how small they are. And kind of even more specifically in what I study, this case study of sorts focuses on a species known as Hypopachus barbari, which, as you can see here, this is Hypopachus barbari, again, relatively small, maybe about the size of a $50, maybe a little larger. And they have this kind of choppy distribution throughout Central America. And the reason for this is because they are a highland adapted species or microhylid. So they only live on mountaintops. They're distributed, again, like I said, across Mexico, Guatemala, El Salvador, and Honduras. And for a long time, there's been predicted cryptic diversity, meaning that there's probably diversity there, but we can't really tease it apart very easily, among populations of Hypopachus barbari. So that kind of brings up a hypothesis of, well, are there actually more species within this species that we really don't know about yet? Which kind of spurned on this study that we have today. So kind of getting into how do we do this? How do taxonomists or how do systematists assess biodiversity or describe biodiversity for that matter? And what we try to do is we try to use a type of taxonomy that's called integrative taxonomy. And traditional taxonomy only tended to use external morphological features. So saying, okay, say the snout is significantly larger on this species than it is this species, or there's less webbing on their toes in this species than there is in this species. And that's basically all traditional taxonomy goes off of. But what integrative taxonomy aims to do is actually use multiple lines of evidence to paint a clearer picture of the true taxonomy of those organisms. So what we end up using is we use that morphology, but we also use genetics. We use osteology, so the bone structure of these organisms. and also other species-specific attributes that may be useful in teasing apart organisms. For example, a lot of frogs, we also use bioacoustics, so their calls, because each call or calls across species can actually differ. So there's other species-specific attributes that we can actually use and integrate into our descriptions of new species or into our assessment of whether a species is different from another one. Another way that we do this is through lots of field work. You can actually see here a couple of the places that we've been in Honduras. This is actually one of the locations that we collected one of our new species of microhylids. And actually, the amount of time that went into collecting the specimens for this study was 10 years of field work and a lot of manpower going into it throughout that time. And what we did was I only, I think, went on 2 of the trips to collect specimens for this. But over that 10-year period, there were specimens collected from most of the highland regions in Honduras where Hypopachus barberi was known from. Okay, so that we could eventually then assess which populations may be different species from true Hypopachus barberi. And actually, the picture here is the type locality of the new species that we described. And what the type locality is it's the area or the region or the literal locality that new species, that specimen is described from. And you can see here this lovely little pond right here, or entrenchment of water, is where that species or that specimen was actually collected from. And you can actually see these, you may or may not be able to see, they're kind of small, but these strings of eggs are actually Hypopachus eggs in the water. And you can see the kind of the beautiful mountains of this area as well. It's a really beautiful area that these were collected from. Another way that we study this is using genetics. So one of kind of our first lines of evidence that we go in with is looking at the genetic makeup of these organisms. And how we do this is we actually extract tissue from the organism, and then we use that tissue to extract DNA from those organisms. And then we isolate and amplify specific fragments or regions of DNA that are interesting to us, that we can assess the levels of genetic diversity amongst the different species that we're actually looking at. And then we sequence those DNA regions that we isolated and amplified. And so when sequencing that, you basically get back your long string of A's, T's, C's, and G's. And then we can align those between all of our different samples. So between our new species, its sister species, Hypopachus barberi, things like that. We align those and we create a big alignment of those genes specifically for those specific organisms. And then we use computer algorithms to build phylogenetic trees or evolutionary trees to determine the relationships and also the diversity and divergence between the different populations, or again, potentially new species that we're actually looking at. And for this study, we actually used 3 genes, 2 mitochondrial genes and 1 nuclear gene. From there, we then use morphometrics, and I kind of already talked about this, and you're measuring different external parts of the body to be able to compare between species. How are they different between species A and species B, or potential species A and species B? And you can actually see up here some of the different measurements that we take. So we take the width of the head, the length of the head, the length of the tibia, the whole leg length, specific head morphologies, or specific head measurements, specific measurements of the hands and feet, stuff like that. And across our study, we actually measured 36 different characteristics that we used in our morphological comparisons between these 2 species. And then we also used osteology, so the bone structure and the bone makeup of these organisms. And the way that we do this is we actually use CT scanning, or computed tomography scanning, which, if you know about CT scanning, basically you can view the internal features of an organism by actually not destroying the organism. In the past, to be able to look at bone structure or osteology, you literally had to basically tear the skin off the preserved specimen and then stain the bones and cartilage using different chemicals to be able to view that. But now we can actually take a museum specimen and then scan it through a CT scanner, and you can see the bone structure, you can see the vascular structure, the neuronic structure, all these different types of structures within there to be able to compare. You can even look at parasites that may be inhabiting the body of that specimen. So CT scanning has really actually allowed this huge boom in being able to look at very specific internal morphologies to tease apart these animals as well. So what did we find from using these different methods? Well, our genetics first came back, and basically we'll look at this phylogenetic tree here just to give you guys a quick kind of rundown of what you're looking at is these areas where these branches connect. So these areas here, basically where you see numbers on here, those are called nodes on a phylogenetic tree. And what those nodes represent is the most recent common ancestor between the two sister species that we're looking at. So if we take this species here, or this group here of specimens, and this group of specimens here, the most recent common ancestor to those two groups is this group here, and so on and so forth as you go out. Well, here's Hypopachus barbari, and here's this potentially new species. And when we actually sequence this, built this phylogenetic tree, you actually see quite a bit of divergence between those two sets of specimens. And so we went, wow, okay, so we actually have decent genetic divergence between these two populations, or between these two potential species. Because before this, all of these would have been considered Hypopachus barbari. So we were like, okay, well, let's continue on based on this genetic data and look at some of these other features, such as morphology. And what we looked at was those 36 different measurements that we took. And you can see these are actually all the same species. They're all the new species. You see a huge degree, or very high degree of variation in color pattern, especially on the vent. You can see this black and white or black and cream kind of modeling on their stomachs. Vary greatly between these species, but they also vary greatly amongst most species within Hypopachus. So that wasn't too surprising. But we did find that our new species actually tended to have longer hind limbs and also had a more pointed snout. So we actually found some more external morphological characteristics that do basically separate these two different species out very well, that were identified genetically as well. And then finally, we use this osteology to look at the skull morphology as well as the whole body. I only put the skulls up here because it's where a lot of it came from. And what we actually find is that there was a lower degree of ossification or bone structure in the skulls of the new species. So here's actually Hypopachus barbari, or our true Hypopachus barbari here. You can see really good ossification of most of the features of the skull. But when you look at the new species here, you actually see that there's a lot less ossification in the bones of the skull. So we said, okay, well, they actually have less ossification too. That's really cool. So we actually have a lot of evidence pointing us towards that these are two different species. And so what did we conclude? Well, we described a new species, and this is this new species here. So these are actually all the same organism, all the same specimen as well. This is the specimen in life, and then this is the preserved specimen that we used to describe it. And what we would call this specimen in the museum that we would collect it from or take it from is called a holotype. And that holotype is actually the specimen that this new species was described from. Okay, and this new description, we called it Hypopachus guancasco. And what guancasco actually is it's a festival that takes place in the region, the Lenca Highlands of Honduras, where this animal is found. And so we tried to use kind of indigenous words or indigenous, give it indigenous meaning to that area that it's found in. And actually you can see here, it's completely endemic to this area of Honduras. And these blue dots right here are the only places that it's found, 2 locations that it's found in. And then Hypopachus barbari is actually shown in orange. And then its other closest group, which is Hypopachus variolosus, is found in green here. But that's the only 2 locations are actually these 2 mountaintops in the Highlands of Honduras. So it is endemic or specific just to that region or just those two mountaintops. So you're probably thinking, well, what's the importance of this work? And I had mentioned that I would come back to this later when I was talking about this in the beginning. One of the biggest reasons that biodiversity discovery is highly important right now is for conservation of these species. So basically what happens is a proper taxonomy, or the understanding of a proper taxonomic relationships between organisms, can lead to proper conservation statuses or concerns being given to these animals. So for example, this species that we just described, which is only found on these two mountaintops, it has a really restricted range and probably really restricted numbers of these amphibians. So it has a higher conservation priority. It was actually described as endangered through this study because of that. And two of the big reasons why conservation is so important in this region of Central America and Central America as a whole is because of two main things that face amphibians in these ecosystems. And one is this fungus, this pathogenic disease that has really devastated the amphibian communities in Central America over probably about the last 40 years, known as chytrid fungus. And you may have heard of it. It actually can be found here in the US. And it targets, there's one species of chytrid that targets frogs and toads, and another species that was probably recently found, maybe in the last 5 or 10 years, that specifically targets and infects salamanders. And biodiversity loss has been driven greatly by this chytrid fungus, especially in Central America. And interestingly enough, some new biodiversity assessments that are taking place in these regions are actually finding some of the biodiversity is bouncing back, which is a great thing, or has kind of renewed itself and come out of being what we thought was extinct or extirpated in certain regions. But another thing that's really driving habitat loss, biodiversity loss in this region is deforestation. So a lot of these amphibians, especially the amphibian that we just, or the frog that we just talked about, inhabits these highland forest areas. And the problem is that the forests are basically being cut down and devastated for logging or things like that. And so basically their habitats are being completely destroyed. So understanding the biodiversity that is in these regions, either before it's lost or while it's still there and existing, is highly important to be able to put conservation concerns or conservation statuses on these regions that could be at risk for deforestation, things like that, so that those ecosystems aren't completely devastated by this. Another thing that comes out of the importance of this work is use of museum collections. So I don't know how many of you have ever been to a natural history museum. When you go to a natural history museum, usually you're like super, at least I was when I was a kid, you're super excited to see like the dinosaur exhibits or the different natural history exhibits that are in that natural history museum. And one of the things that we either don't know or that sometimes we forget is that kind of behind the walls of those exhibits that we look at are actually these huge repositories of natural history data. And you can actually see this here. This picture is actually from the Amphibian and Reptile Diversity and Research Center here at the University of Texas Arlington, where I work. And it's only reptiles and amphibians, but most other natural history museums, large natural history museums like the Smithsonian or the Carnegie Museum of Science in Pittsburgh, or the Carnegie Natural History Museum, excuse me, in Pittsburgh, places like that, the Cal Academy of Science in California, they actually have these huge repositories of animals and plants kind of behind closed doors that are used for biodiversity discovery and biodiversity studies like this one that I'm presenting here. And it can allow researchers to look at different biodiversity in regions. It can also allow them to look at rapid adaptations of species, being able to look back in time. A lot of these specimens date back sometimes to the 1950s, and actually even further back in time. If you're at a larger institute, that can go back into the 1800s when people were collecting species like that. And often too, a lot of these museums have tissue collections that are associated with them, so that we can use them also for molecular studies, which is extremely helpful. And actually even more now, scientists and systematists are forming ways to actually be able to extract DNA from preserved specimens that don't have tissues. So in the next couple of years, it's going to be really interesting to see more and more how museum collections are actually to be able to be used for these biodiversity assessments in the long run, and species discovery assessments. So that's kind of just a snapshot of kind of a really small part of my research. So I wanted to just kind of give you guys a taste of kind of what else I do. And kind of my research falls under this idea of biodiversity genomics. That's why I work in the lab that I work in. It's basically a biodiversity genomics lab. And we kind of study the interplay between organismal biology and natural history, evolution, genomics, all that stuff. And kind of, like I said, it meshes together into what's known as biodiversity genomics. And I kind of have three things that I study outside of this. One is looking at Central American amphibian diversification, along with this, the taxonomy of these things. And so, the taxonomy only can tell us part of the story about these species. Obviously, it tells us, well, species A is different from species B. But one of the things that I'm very interested in is, well, what drove species A and species B to become two separate species? So I'm very interested in kind of the processes that are at work to allow these organisms to diversify. You can see some work here that I've done in toads in Central America that has been really cool, seeing how highland diversification has allowed these three species to basically diversify. One of the other things that I've done is, since I've been in Texas, I've also worked on conservation and systematics of Texas reptiles, one of which being the greater earless lizard, which you can see here. Holbroechia lacerata. And what happened here is that Holbroechia lacerata had two subspecies underneath it, Lacerata lacerata and Lacerata subcaudalis. And it's been argued back and forth whether or not these are actually two species, whether they are or not, so on and so forth. And so part of a study that I was part of looked at and assessed the mitochondrial differentiation between these two subspecies, and we actually see really, really, really deep divergence between these two separate species. You can see one species here and one species here. You can also see different morphologies in patternation on their legs, things like that. And we assessed that they are two separate species. And we've also worked to do some other conservation-related work with the species, because now the new species, Holbroechia subcaudalis, is actually highly extirpated from certain regions of Texas now, and also highly threatened due to habitat loss and habitat change just within Texas here. And then kind of the last thing that I study that I always am really excited about, because it's something new that I've come into studying, is actually the evolution of toad toxins. So you guys have probably all run into toads that look like this guy here. You kind of find them all over the place. They're very generalized species. But if you didn't know this, they're actually toxic. Don't go out and lick toads is basically what I'm saying. Otherwise, they're perfectly fine to handle. But it's really cool. These things actually are able to synthesize their own toxins. One specific one is cardiac glycoside that I'm very interested in how it is actually synthesized by these animals. And the really other interesting thing, and kind of even the broader evolutionary context of this, is that toads are only one species kind of in across the tree of life that actually synthesize these cardiac glycosides, because there are actually other animals, such as insects, beetles, and fireflies actually synthesize them. But even further kind of along the tree of life, there's numerous species of plants that actually also synthesize these toxins. So I've become very interested in kind of the evolutionary processes that are at work for how plants and insects and toads have all come to the same conclusion or converged on this evolutionary trait by which to synthesize these toxins within their own bodies. So it's kind of an ongoing process, an ongoing project that I've been working on with this as well, and just find absolutely fascinating. And at that, thank you guys so much. And if you have any questions at this time, please, I'll take them.
Thank you, TJ. That was great.
Thanks so much, Leah.
It's really just fascinating to hear that you can spend a lifetime studying just frogs alone.
Right, and it's great too. Who doesn't want to study frogs?
Yeah. So my first question kind of goes right to that. How did you end up studying frogs specifically?
Yeah, so where I grew up in northeastern Pennsylvania, up in the Poconos. And it's kind of funny because the Poconos is part of Appalachia. And for those of you who don't actually know this, it's a kind of a really interesting fact. The Appalachian Mountains actually have some of the highest salamander diversity in the United States. And actually, it has the highest salamander diversity in the world, surprisingly enough, which is kind of amazing. So it's this huge hotspot for salamander diversity. And growing up there, of course, I was always outside in nature, flipping stones, flipping rocks, coming across newts and salamanders, and also frogs. So kind of growing up in that, I was always interested in these organisms. But then when I went into my undergrad, I actually started to get into some genetic research on toads specifically. So I've stuck true to toads for a long time. But it just kind of fascinated me, the kind of evolutionary questions that you can ask from these organisms that you just wouldn't even think about most days. There's just such a plethora of questions that you can ask across the spectrum of evolutionary questions, systematic questions, things like that. So it kind of, having been in that atmosphere of being able to see these things firsthand throughout my childhood, and then being able to say, oh, cool, I can actually work on these as a career, made it all the better, and kind of is how I've pressed on with using them as model organisms and making a living out of studying frogs.
Interesting. So I want to go back to some of the research that you talked about. And you were talking about how once scientists discover a new species, they're able to kind of separate out what genetic traits make them different and how they look different and all those things.
Right.
How do they first discover a new species?
So typically, in the past, it's usually been due to morphological differentiation. You measure these things and you say, oh, wait, okay, in comparing these things, this thing is a lot different than this, or even sometimes subtle significant differences. These two things are different. But in the age of genetics and genomics, it's made it a lot easier to just take your piece of tissue, extract DNA out of it, and within a couple of maybe 2 or 3 days, you have basically the genetic code for this thing.
Mm-hmm.
And so genetics has really changed it, where you just basically what we do what's called barcoding. So you amplify one single gene or region of a gene. It's called CO1 or 16S, part of the mitochondrial genome. You barcode those things, and then you can make a huge database of all different species that are known. And you can then see where some species have actually diverged from each other and say, oh, wait a second, these two things are actually very genetically different. We should probably look at these and assess these using morphology or using osteology or bioacoustic, stuff like that. So really it's shifted from just observational looking at kind of morphological features to shifting sort of towards the genomic and barcoding side of things, where we can say, oh, okay, these things are genetically different. Let's assess them further. And that has helped immensely in pushing forward biodiversity discovery and making it more rapid as well.
Mm-hmm. Interesting. With technology, I think it sounds like you're saying you can now prove these changes, which have only been observed in the past.
Right, exactly.
Fascinating. So back to your research about this specific species, Hippopotamus berberi. Mm-hmm. What does research for you look like when you study something that's only located in Central America?
Do you mean under the current conditions, or like?
Well, I think in the life of a scientist, when you study something that isn't where you live, are you taking trips there, bringing samples back? What does that look like?
So typically we would fly down there, do our field work for X number of weeks, collect what we need to. When we collect the specimens, we obviously preserve them, and then we also take the tissue samples. And then when we come back, we ship everything back with us. That means the specimen, the tissue, everything comes back to the lab with us here. And then we do all of our genetic work here. We then also use the preserved specimens to measure or do the CT scanning on, stuff like that. And then we also deposit those specimens into a museum somewhere. So a lot of the specimens that we used for this study are from Florida, University of Florida Museum, the Carnegie Museum, and then luckily the museum that I mentioned that's here, the ARDRC at UTA, actually has some of the largest numbers of Central American herps in the museum alone. So we were able to get a whole ton of specimens to be able to do the morphological studies right here, basically in my backyard, so to speak. from the university and got those on loan. We're able to do what we needed to. So really, the field work is always awesome. It's great trips to be able to go down there and be able to basically integrate yourself into the country as well as the ecosystems down there. And it is a lot. It's a lot of work. I mean, that if you're down there for 4 or 5 weeks, you're traveling constantly from place to place to place, or from field site to field site, and it's tiring. And then you get back and you sleep for a little while, but then you just hit the ground running and do the actual lab work from there. And that's sort of why I've shifted a little bit of my research more into North American taxas, because it is easier. And the great thing is that the diversity that I've been studying, there's tons of diversity right here in Texas as well. So there's plenty of diversity to look at in the U.S. as well that hasn't really been looked at. So it really helps, obviously, not having to travel as far.
Right.
It's always great being able to go back down to Honduras and do field work down there.
And in the context of the pandemic, we were talking about this before the live broadcast. It sounds like through the sharing of resources, you're still able to do some of that research without the field work.
Absolutely. I mean, one of the biggest impediments right now is getting specimens from museums, because a lot of museums are either shut down or they're not. I guess don't want to say not willing to send you specimens, but they're not able to send you specimens right now. It's funny because sitting on the desk behind me, I actually have the specimens from the study that are from Florida and from Pittsburgh that we got, and they've been sitting on my desk since March. And actually, I've had them for probably about 1.5 years now, which is way over the loan time. But just trying to return them has even been difficult. So doing some of this work has been difficult during this time. Luckily, we had all the data collected for this project prior to the pandemic. And actually, this was my writing this all up to go out for publication was my first pandemic project, so to speak. And we actually just had it accepted. It'll hopefully be published by the end of the year.
That's fantastic.
Yeah.
Wow. So if you're watching and want to know more about that, please stay tuned. We do hope that you get published really soon.
Yeah, we'll see.
So pre-pandemic, when we're talking about your fieldwork in Honduras, how common is that species in the area, and what does researching them look like? Do you have to hunt for them to actually be able to then study them?
So you have to go there at the right time of year, that's for sure. And it's funny because in 2015, it's the only time that I've ever actually seen this species. And the fieldwork pictures that I showed you guys is where we actually were when we found them in 2015. And it was funny because we just happened to be looking, I was mainly looking for toads, was not thinking about this species. And we were also looking for some salamanders, and one of the undergraduates that was in the lab with me was like, hey, I think there's something in this like little tiny, I mean, literally like little tiny pond thing. I don't even know if you can call it a pond, but basically this entrenchment that had water in it. And she's like, I think there's something in here. And so I ran over and we were both looking, and we scooped out two specimens of Hypophagus. And when we finally got back to the field site with Joe was like, oh my God, I haven't seen these since 2000, whatever. I think the last time he collected something was like 2009 of these things. Because like I said, they live this very fossorial lifestyle. So they're underground for most of the year. They come out for a week or two, breed, eat, and then go back to sleep, so to speak, for a long time. And it was funny because, again, kind of a pre-pandemic, actually not even pre-pandemic, last year, I went back down to Honduras with Dan was there, and with Joe, another team of undergraduate students. And we were supposed to go and hopefully collect call data for these species, or these Hypophagus barbari and Hypophagus guancasco, and some other stuff got in the way. Some of the political instability down there got in our way. We had to totally change the trip for that matter, and we missed them, so we didn't get to go back. And it was funny because as I started writing the paper and everything, like I said, kind of right before the pandemic really hit, Joe has been down in Honduras for the year. And he was like, well, I'll get out there in April when they start to breed, or when they usually start to breed. And then, of course, the pandemic hit. And he's like, there's no way I'm making it to the field. Sorry, man. And it was like, no, totally understandable. But there's things that get in the way. And like I said, 10 years of field work went into collecting, I think, only 17 specimens of this new species.
Wow.
So they're really difficult to find in the long run. So that's what does make this difficult, especially working with this group of frogs. There's other frogs that I work on. So like the toads that I work on, it's great because they're so common most of the time that we can go out and we basically do what's called road cruising. And we drive up and down the roads at night after it's rained, and you look for things happening across the road, and you just get out, toss them in your bag, get back in the car, and keep driving. That's how easy they are to find them. You can find 30, 40 in a night, whereas these guys you find 17 in 10 years.
Wow.
So yeah.
Very important research.
Yeah, absolutely.
And if you're watching and you are so curious about this, you're also going to get to hear more in a couple weeks. We will have Joe Townsend joining us from Honduras. So stay tuned for that. We're going to wrap up soon, but TJ, I want to leave some of our young watchers with some inspiration here. If you are watching and maybe you're interested in amphibians or frogs specifically and would like to potentially have a career in that one day, what would you recommend for a young person as far as educational path?
Well, obviously, probably the best educational path is get your undergrad degree. And from there, when you're working on your undergrad degree, try to work in research, see how you like it. One of the great things about working with amphibians and reptiles and evolutionary biology, conservation, things like that, there's a lot of career paths. You can go into fully working in conservation. You can work in doing mainly field work. So as an undergraduate, I got to work on, though it wasn't reptiles and amphibians, I actually worked on a lot of fish surveys for the Pennsylvania Department of Environmental Protection. It was probably one of the best jobs I had, especially as an undergrad. I think I was 21 when I did that. And it taught me a lot about field work. But it also showed me, you know what, you don't have to have your PhD to be able to do this really cool research and stuff. Because most of the people I worked with had their master's or had their undergrad. So obviously going to college and kind of figuring out the way that you want to go about it. Do you want to continue to do research? Do you want a tech job or biotech job, things like that? You can still work on the things that you enjoy without just doing research. So there's a lot of kind of find your niche and find what you really enjoy during college, and then go from there. Really kind of the way to go about it.
Well, there you have it. Inspiration from a real frog scientist. TJ Firneno, thank you so much for being here with us.
Thank you so much for having me.
You are very welcome. Before we wrap up, I just want to thank the Nevada County Relief Fund and Placer County for donating to our online learning programs, helping us keep them free. If you're watching and want to keep these programs free, you can always donate on our website as well. And we will catch you next week. Thanks, TJ. Have a good day.
You too. Thanks so much, Leah.